Hello guys, my name is Dr. Varun and you're watching Chlorophyll classes only on YouTube. Well, if you know that this is another day and it's the day for the 12th class, and today we're going to be starting with a new chapter. But this new chapter has an essence from the genetics unit which we have covered so far. So first we're going to be doing this understanding that what is about the genetics, and then only you go on it, then certain pointers which might not be able to understand directly, you can understand it now, and I will try to make you connect with it. So let's just dive into it. So biotechnology, the name it says that biology and technology, there is a union of these two things. So basically biotechnology is what? Nothing. It deals with the techniques using the live organisms, their parts, certain of things to obtain the product and the processes which are useful for humans. Means, for example, if I want a particular medicine, maybe I can help a organism to make it for me. That is called as biotechnology. Or maybe you might have studied about the microbes in human welfare also, where you studied about that certain curd is being made, milk we are getting. These are what? These are the curd and the penicillin and all those things that are microorganisms forming. It's a traditional way of using the biology to obtain the things from it through the technology and the understanding of the concept. Modern biotechnology utilizes the recombinant DNA, which is our major thing to study over it. Let's just write about these things. So biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful for humans. They are of two types. What are the two types? Number one is the traditional or classical. Another is the modern or new. No, the thing is that in traditional, what is happening? Uses natural capability of our organisms to obtain the product from it. Okay, uses natural capability of the organism. Example, curd from lactobacillus, penicillin from Penicillium notatum. So this is what traditional way of it. Now what we say that this is the casual life. What is Mentos life? Mentos life is like we will change the DNA itself. Okay, so based on recombinant DNA technology, what it is, how it is, this is what our chapter will be. Okay, so now if someone asks that what would be the definition of it, so we will write that the integration of natural science and organism or the part or the cell of an organism even the molecular analog, like your DNA is being a recombinant, so means we are using the certain part of the DNA. Okay, so molecular analogs for product and service. Product and service, nothing else, just the fancy way of telling this thing. Now, who is called as father of biotech? Father of genetic engineering. Father of genetic engineering engineering, that is known as Paul Berg. What he did? So he took out the plasmid from the Staphylococcus, okay, and put it inside the E. coli. Now the E. coli is able to get certain features which were present on the plasmids of Staphylococcus. This is the history of it, which is given in your NCRT as well. So you will be remembering this story. Okay, now if someone has to do the biotech, so there must be certain principles on which it has to do it. So there are two principles. One is the genetic engineering, means you are making the recombinant DNA technology, and the second is the bioprocess engineering, means you are able to maintain an environment to produce it multiple times for the human consumption. Okay, so that is what the two important principle of biotech. Okay, so principle of biotechnology. Meanwhile, if you're liking these videos, give me a like, share it with your friends, because boards are there and sharing is caring. Moving on to the next thing, that is, if you are feeling that this classes are not enough, you want one-on-one mentorship with me, your doubts to be there, all the syllabus to be done properly on time with a proper schedule, then you can look forward for my board classes for 12th class. If you are in 12th and you want to give your NEET and want to score 360 out of 360, then also you can join the classes. We have a course for a NEET 2026 also for the board students plus the dropper students. You can check it out on my Instagrams. Links will be available to you, or you can simply check it out chlorophyll underscore classes on Instagram for the rates and all the details about the program. Okay, so the principles: number one, genetic engineering, or it is called what? Recombinant DNA technology. Now in this, what is happening in this techniques to alter the chemistry of the genetic material, that is, we are changing the DNA or the RNA and then introducing it into an organism and then making that organism have that changes sustained in the next generation also. So this whole process is very important for us. Okay, so this is what makes the crux of the process which we're going to be studying in this chapter. Okay, so it is what we need to know, the techniques to alter the chemistry of genetic material, DNA or RNA, then to introduce these changes into the host cell, to introduce these into a host organism, thus changes the genotype of the phenotype of the organisms, because it will give the new characteristics, new things you have been added, new characteristics will come into the organism, thus change the genotype or phenotype. You know what is the meaning of these words. Okay, phenotype is a physical appearance may be changed, genotype means genetic makeup will be changed. Okay, of the host organism in which you have added it. So for example, if I am the host, you take out the DNA of a jellyfish and put it inside me, and now I will be having what certain characteristic of the jellyfish was into me. How that happened, that we will learn it later. Okay, organism, and this organism is called as genetically modified organism. So if anyone over here is the Marvel fan, then can we say that not the Superman is not there, it's DC. Is the Captain America a type of a modified organism? Yeah, he is genetically modified one. This is the first thing. Second is what? Bioprocessing engineering, means the process to maintain it and to make it available for the mass. That is also important. So the second thing is bioprocess engineering. So in this, what happened? That is maintenance of sterile. Sterile means microbial free. It is not that sterile that you can't reproduce. Okay, microbial contamination free environment to enable growth of only desired cell in large quantity, because that only going to be making what? Our product of interest. That's it. So the next question will be what are the main basic basis of the DNA technology? It is what? Making identical copies of any template of DNA which we want it to be keep on translating, which is important for us because it's containing what? Genes which we wanted. Okay, so that is the main basis of the recombinant DNA technology. So here we're going to be writing it up. If someone will ask like what is the main basis of recombinant DNA technology, to be able to make multiple copies of multiple identical copies of any template of DNA. Now if we have to sum up the whole process into 3 steps, what would be the step? First of all, we will identify the DNA with the suitable desired characters or genes which we wanted. Then second thing will be what? Putting it into the host, whatever the host, you put it into it. And third is maintaining this into the next generation also. So if you have given me the features of jellyfish, which I gave the example, if you give me a feature of a jellyfish, then my child should also have those features present. So these are the 3 most basis of the process. Okay, so if I have to write that 3 basis of, or 3 basic step, 3 basic steps. Of course, in reality, there are multiple steps, but here the basic steps we are trying to reduce the process in understanding format, okay, or the digestible process. First is identification of DNA with desirable genes. Once you have identified, next would be what? Introduction of the identified DNA into a host. And third is maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Things which we have discussed, we have told up here. Now, if someone will ask like, okay, for a biotechnology to be a successful and why we are studying it, there must be certain things. So 2 most important discoveries which leads to foundation stone for this branch and this unit in our syllabus is the presence of plasmids and the endonucleases, restriction endonucleases. What are they? We will learn it one by one, but here we're going to be writing these two things, that two most important discoveries which lead to foundation of biotechnology are: number one is plasmid, number two is restriction endonuclease. What it is, how it is, we gonna be learning all these things. Okay, now let's just understand that what the whole process will be doing. Means this thing I'm gonna be helping you into making it a little slowly enlarged form so that you can digest it. Till now you know the basics of it. Let's just see that how we're gonna be doing these things. Okay, now in our chapter we are doing an indirect way of doing this biotechnology, not the direct way. There are direct ways also there, which at the end we will see it, but here in your book they have tell you what indirect way of doing the things, few things. Okay, so let's just start with it. So first of all, you know what, you need a desired gene. For example, I am a very not a dentist. Oh god, if my friends gonna ever able to see this video, they will be like gonna kill me that how you calling the field psycho and all those things which you are actually studied. But no, it's not like that. Coming back to the natural thing, to our study, that is that if I'm a psychoscientist and I want to make a bioluminescence feature in the goldfish, okay, so means in all the goldfish shells it should shine, it should glow in the dark. Okay, this is my target. Now I need certain genes which can actually do that. So right, from where I will get that? So I know that jellyfish has a feature of bioluminescence which belongs to Ctenophora, right? So I will get that jellyfish. So my first thing is what? I get that jellyfish. So this is my jellyfish. This jellyfish has what features? This jellyfish can glow in dark. Okay, so this is the feature. So I am showing it that it's a glow feature. So this is what it's glowing. Certain of its tentacle also glowing. Okay, so this is what glowing. Okay, so this is what bioluminescence feature. Now this feature would be there because of certain gene present. Gene will form RNA, RNA will form the proteins, and proteins will make it glow. Right, this is only happen. So understood that molecular basis. So if I took the gene will do all the rest of the work. So I want that gene to be it. So what I did human genome project. Okay, and in human genome project I am able to find the gene from it. Okay, so I'm able to extract the DNA of it. So first step would be what? Extraction of DNA. Extraction of DNA. So here I am showing the DNA to you. So this is my jellyfish DNA. So this is what? Jellyfish DNA. Now in this DNA, you know that certain part only will have that particular gene. So after human genome project, same genome project, jellyfish genome project, we are able to find that at this place, okay, at this place, my gene of interest is there. Gene of interest is there. What is the interest? Bioluminescent feature. Okay, so this is gene of interest present. So what I did, I used an enzyme, endonucleases. Okay, restriction endonucleates. I cut it. So cut it out with help of restriction endonucleases. So now in my hand, what is there? This gene is there. It shows what? It has the property of bioluminescence. Okay, so it has the property of bioluminescence. Now I want that this genes to be present inside the cell of the cell of the fish. Okay, each cell should have it, or else all the cells will not glow, right? I want the whole of the body of that grow. So now I have to put it inside. But directly if I put anything, will the cell take? No, the cell will found it like, okay, this is foreign, let digest it and destroy it. So we had to make the host cell to take it. So here we need what? Delivery agent. So who will be our delivery agent? Swiggy, Amazon, Flipkart, whatever you want to take it. I am calling it plasmid. So plasmid would be my what? Delivery agent. Okay, so I will mask this thing on the delivery package and send it. It's just like sending a Trojan or sending a virus inside the cell. Okay, so first let's see that our plasmid. Okay, okay, 1 second. So this is my plasmid. Now this plasmid will have what? This plasmid will, if it is circular plasmid, okay, this look like this. So I'm using what? Vector, which is what? Plasmid. It is also a what? Genetic material only. Okay, what it is we will learn it. Okay, so I took the plasmid. It's a genetic material. It is an extra stretch of circular DNA present inside what? Inside the bacteria. Okay, certain bacteria I have taken. From here I took this plasmid of it. Now since I have to add this sequence into it, how I will add it? First I will cut it like normally. If you want to add an extra page, okay, just imagine that, for example, if this is the thing, okay, this, okay, and I want to join it, anything, what you will do? You will put the glues and join it, right? Same over here. But if I want to add it over here at this one, so first I will break this and then I will add and extend it, right? So here also what will happen? This plasmid, first I will cut it. Okay, this also I will be cutting with the restriction endonuclease, similar restriction endonuclease. Why similar? That I will tell you later on when we were doing the things. So I will add that. Okay, let's just do it. So I will cut this also. So over here I made a cut. So this is what? Also cut out with similar restriction endonuclease. Now after cut out, how it will look like? Let's see. This way. Okay, rest all the sequences are there, just certain sequence has been cut, nothing else has been changed. Okay, so rest is there, bonds are there, these bonds are opened up. Okay, now what will happen? At this area only, this thing will go and attach to it. Okay, so let's just see that. So the next step is joining of these two segments, joining of these two, okay, plasmid and the gene of interest. So let's just make it. Can you see this? So the thing that has been made up, this is what this is called as recombinant DNA. Because plasmid is what? It's a vector DNA. It's a DNA part stretch of the DNA only. But since it has taken the new thing, the yellow color thing, which is what giving the feature of bioluminescent. Okay, so this become recombinant DNA or recombinant vector DNA. Okay, now what we will do, we will put this into the zygote. Now major question is zygote of whom? Zygote of the fish. Why I took the zygote? Because zygote is what? It is an undifferentiated cells. Means it, and it has a capacity what? Keep on dividing. Sexual reproduction in human beings, you know na? Same thing happen in all the organisms. That zygote will divide and form all the cells. Initially the zygote also look, zygote, embryo, morula and all those look similar in the cells only. Okay, so what will happen? Now I will put this into transfer it into zygote of the host cell. Why host cell? So that because the host will only show the effect na, that the fish. So I put it into the fish zygote. So this is the zygote, and when this zygote will grow into adult, what will happen? For example, this is the fish. Okay, this can be better. Fish is there, right? Now what is the feature that every of this fish shell will have? The fish will glow. Okay, and the glowing reason is what? The reason is your biotechnological efforts that has been made. So all of this will glow. So me being the scientist who is very much crazy, fish has been obtained. Does it really happen? Yes, some Japanese scientists did that. Okay, now we're going to be studying about the tools required for biotechnology in this video, and in the next video we're going to be seeing the process, how it takes place. So let's try the tools. So 4 different major tools we required in it. Inside it multiple things are there, but these 4 are the broad classification. Okay, so the next topic is tools required for biotechnology. And first we're going to be studying about these tools only, and then we will understand its working, how the whole scenario is being done. So 4 tools are required. Number 1 is enzymes. Number 2 is vehicle DNA, or we call it vector. Then we need what? Desired or alien or passenger DNA. And the last one is the host cell, in which we have to put it in. So if you remember that just we did what? The flow chart of it, we see that where these enzymes would be there. Okay, so if we go back, so then you will see that the enzymes like this endonuclease, joining of these two, here enzymes will work. Okay, then we ask that the vector or vehicle DNA. So this is the vector or the vehicle on which we put it this. Then the desired DNA, desired segment of the DNA. And the last is the host. This is the host cell in which this whole structure will be present in each of this cell. So these are the 4 important pointers of it. Let's just study one by one, starting with the enzymes. Okay. Now there are 4 enzymes that we're gonna be studying. What are these 4 enzymes? The 4 enzymes are as such. We have lysing enzyme, we have cleaving enzyme, after that we have lysing enzyme, cleaving enzyme, ligase enzyme, and one more we needed. What was that? Synthesizing enzyme. Yes, synthesizing enzyme. Let's one by one study about them, starting with the first, lysing enzyme. So lysing enzymes are what? They help in breakdown of the other thing except the DNA. For example, from that jellyfish, inside jellyfish cell, DNA will be present where? Inside the nucleus, but outside there will be a cell membrane. If we are taking it from the plant, in case if we are taking the plant feature, then the plant cell wall will be there. Inside lot of material will be there. So we have to digest, remove all of that thing to reach to the DNA part. Okay, so that's why the lysing enzymes will help in work. If you remember, this is the same thing we have studied the concept in the biochemical characterization of the enzymes. So understood that thing only, that we will be removing all the other thing, digesting all the other things, and only left out with what? The DNA. So that is what the lysing enzyme. Lysing is what? Breaking enzymes that will help us. Okay, so starting with the lysing enzyme. And the name you have to remember which one is there. How to use it, we will let you know. Okay, so these enzymes are used for opening the cell to get DNA for genetic experiment. For example, lysosomes are used to break bacterial cell. Same, cellulase and pectinase for plant cell. Similarly, chitin is where present? It is present in the cell wall of fungi. So there it will be used. Chitinase for fungal cell or fungi cell. Understood? This is the lysosome. Okay, so this is the first one. In this is the first. Okay, lysing enzyme. After that comes the cleaving enzymes. Okay, so second is cleaving enzyme. Cleaving enzymes means what? They will cut. Okay, so they're used to cleave the DNA. You know that we have to cut the DNA of the plasmid also. We have to cut the DNA of the gene of interest place where it is present. So at these two places we have to make the cut, and we have to make the cut. Who will do it? Certain enzymes only will do it inside the cells. So these cleaving enzymes are called as molecular scissors. Okay, so they are what? They are molecular scissors. So what they do? They are used to cleave / cut DNA molecule. Now there are two types of cleaving enzymes. One is exonuclease, other is endonuclease. So two types. One is exonuclease, another one is endonuclease. Now exonuclease one. Okay, the exo means outside, exterior. So here what happened, these remove the nucleotides or cut the segment as the extreme end of the DNA. So means, for example, if this is the DNA, this is the DNA, and same I am making over here also, so that I can tell you what is happening. So what they will do, they will cut it like this here. So what happened, this end area is only being cutted out, but maybe your gene of interest is not present at the end, maybe it is present here. Then what? Most of the time it is present in between anywhere. So you need to cut in between somewhere. So endonuclease will do that. And endonuclease is what? That is our main important to understand about it in a detail. So exonuclease what? Removes nucleotides. Removes nucleotides. Cut at end. Cut at end. Here what happened? Cut at mid or in between. Okay, so remove nucleotide. What is this? In between. Remove nucleotide from the ends of the DNA. Whereas endonuclease makes cut at specific positions. These specific positions are called as, already it is very much determined by the nucleotide, that this nucleotide will cut it here only. Okay, which is called as what? Restriction sites within the DNA. This is called as restriction endonuclease because they are restricted for a particular space or a site to cut. Okay, so these are what? These are restriction endonuclease, which is also called as what? Molecular scissor or knife. It is also called as knife because it is doing what? It is doing the work of cutting. Okay, they are normally what? 4 to 8 nucleotide sequence. So it is a sequence of 4 to 8 nucleotides. Now these sites are palindromic. Okay, so they identify palindromic sites. Usually, sometimes cannot be, but usually it is. Palindromic means what? From one DNA strand or the other DNA strand, it will read same. Okay, means for example, Malayalam. So whether you read it from this or you read it from here, it will spell same. And that's the major thing. Means if there is one endonuclease, okay, this is also similar endonuclease, it is also endonuclease. Okay, because multiple of these molecules will be present, it goes and cut at A and L. So over here also it will go and cut at the AL. Okay, so this thing is what is being done by it. Okay, let's make it like this. This endonuclease will cut at this point. So what will happen? A similar side on both the strand it will cut. Of course, this Malayalam I am showing you like that. Imagine that what? There is what? DNA molecule present over here. Let's make it. One DNA is like this, other is like this. So it will cut what? At a specific sequence. Okay, so we understand. Now, from where do we get this thing? Okay, endonuclease. So next question is, from where do we get this endonuclease? Means it is cutting the DNA, na? So it must be we are getting it from where? So the answer to this question is from bacteria. Now the question is, bacteria also has its own DNA. Why not those endonuclease cutting the normal DNA of the bacteria? Now we understood that if the bacteria DNA will cut, it will what? It will destroy the molecule. So people will say that no, it won't cut its own DNA. But how? There must be certain protective mechanism. Yes, there is a protective mechanism. What happened? That the DNA of the bacteria is what? It is methylated. Means it has a covering of methylated. Because of that, it is being protected and not being cutted out. Okay, so but then why this thing is even present in bacteria? Next question. It is present in the bacteria because bacteria sometimes get infection, and in that infection, if you remember the Hershey and Chase experiment, bacteria was there. It is being infected by the bacteriophage. Bacteriophage releases its DNA and then it started the infection. If you remember that, if no, go back and see that genetic experiment. How do we find out the genetic material is DNA? Topic on my channel only. So you would understand that the bacteria is this has been infected by the genetic material of outside bacteriophage. So what will happen whenever that bacteriophage will release certain things? The bacteria own immunity defense mechanism will do what? It will cut it out and protects the bacteria from getting infection. Understood this thing? Let's just write it out. So next question is, what is function of endonuclease in bacteria? So the answer is they cleave / break the viral DNA. Virus is what? Bacteriophage. Okay, into fragments so that it can save themselves from bacteriophage. Next question is how bacteria save its own DNA. Okay, how bacteria saves its own DNA. The answer is by methylation of its own DNA. Okay, so it protects itself, it is not being identified. Now the next question is, which is the first endonuclease that has been identified? So the first endonuclease that is identified is HIND2. Now the question is that if it is the first, why not the HIND1 name? That also we will come up. Okay, so it has what? It identifies 6 base pair. So this 6 base pair palindromic sequence is there. So that is what? GTC, that is GAC. So opposite will be GTCGAC. So where it cuts, so it basically cut at this G and C point. Okay, so you have to remember that cuts at guanine and cytosine in between. We will learn that what is happening over here. But how many such nucleotides we have identified? 900 such has been identified from the different 230 different strains of the bacteria. Okay, so today it will be more than that we will understand. Okay, today 900 endonuclease has been identified from over 230 different strains of bacteria. Next comes the nomenclature, where we will understand that why did we set that HIND2 not HIND1, but if it's the first. Okay, so nomenclature of enzyme. So see this. For example, this is eco R1. Okay, this is the one such enzyme. Okay, so in this enzyme, this first alphabet, okay, the first letter, it should be in capital, represent the genus of the bacteria, represent the genus of the bacteria. Here it is what? That E. coli we have obtained from E. coli. So E is what? Escherichia. Okay. Then comes the next thing. This CO. Next two words. Okay. So second and third word. It represent what? It represent the species. It is written in small. Written in small. Written in small. Okay. So this is what? Example, the species is coli. Escherichia coli. Then R is representing what? The next thing. This R is representing, it has to be capital. Okay, so it is representing what? Stain. Fourth letter representing strain of the bacteria, and it is completely optional to use. And the last one, this one, this is not I, this is numerical. Okay, it represent what? It represent the order in which, first of all, it is written in what? Roman font. And it represent what? Roman font means 1 2 3, like we write like 1 2 3 4 like this. These are what? Roman numericals. Okay, so this numerical we have to write it in that way, and it represent what? It represent the sequence in which any enzyme is extracted from the body. Okay, means it represent what? Sequence or order of the enzyme extracted from this particular species. Means it is the first enzyme that is being identified in the Escherichia coli. Means when we come to this part, if this is the first endonuclease, they are not talking about endonuclease. Endonuclease name is this, but this is not the first enzyme. We get it from the Hemophilia influenza D strain, and this the second enzyme is the by chance endonuclease. Understood this thing? So this is what the nomenclature, which is super important. Next come the restriction enzymes. When they cut in the two different ways. Okay, and let's see that which one is important for us. Okay, so next pointer that we're going to be studying is so what are the two types? One is the sticky ends and one is the blunt ends. Sticky ends means something would be sticky. That's why it is much easier for it to join. So if you look over here, okay, this when we have made this, can you see that if there would be already certain glues present, it can easily join over here, that glue, and can add what? The things which we wanted, right? So this is the major point. We want the sticky ends. So we have understood that why we need sticky ends also. So sticky ends would be how we will generate it. If, for example, if this is your gene, but certain extra stretch has been added, just one of the strand. So the complementary of it is missing now. The complementary thing that would be missing will help what? It will easily be finding the other complementary part, which will be present on the vector, which should be present on the vector. And because of this reason only, we take the same nucleotidase or the nuclease, okay, enzyme. Let's just see one by one and you will get it. Okay, so sticky end cuts the strands of DNA a little away from the center of palindrome site, but between the same bases, like GC, okay, same bases on opposite strands, which leads to what? There are certain single stranded portions at the end. Leaves single stranded portion at the ends. And these overhangings, like for example, imagine that this is the sequence. So if you see this part and this part is extended part, and this is the gene. So the thing that is paired, this area is paired, this is our gene of interest, and this is part is what? These two parts. This is the overhanging stretches, okay, or called as sticky ends, okay, or overhanging stretches, whatever you want to call it. And these are what? Since these are the complementary, they will be all the time will be finding like zombie that its next part, its complementary part should come and join with it. So the next should be what? The complementary of these should be present on the vector also. So that kind of vector also we have to find. So most crucial point is the human genome project, okay, or the genome project of this and that to mix up and coming together. We will understand this slowly. Now they named it because it has what? Hydrogen bonds, which will go and directly wants to join with another complementary part of it. Okay, so they are named so as they form hydrogen bond with their counterpart. If I have to show you the example, let's say the example of E. coli, ECO R1. Now the ECO R1 will cut your DNA fragment at a particular point. What is the point? Like for example, GAATTC. This is the one strand. The other strand will have what? CTTAAG. So this E. coli thing cuts at GA. So GA point. So what it will form? It will form G on this, C here, TTAA, okay, plus there will be segment of what? This, the upper segment AATTC and G over here. Can you see that this counterpart of it is unavailable? Means what happened? that you can add certain part here and here of it. Okay, and this is the sequence over here would be there. This is the counterpart of it, which should also come over here. So understood this thing, that the cuts are being made and how it is being used up. Okay, coming back onto the blunt ends. In blunt ends, what happen? In your blunt ends, these are also considered as non-sticky ends. Non-sticky ends, which do what? It cleave the DNA, both strands at the same nucleotide position, This results into what? Blunt ends. Results into creating blunt ends. Example, Hind2 that I told you. What is happening in Hind2? It identified GTC, GAC. Opposite will be CAG, CTG. Okay, so it cuts at what? At this level. So which leads to what? It leads to formation of GTC, CAG, one strand, GAC and CTG. This is what? Blunt ends. Nothing can have a counterpart of it, and if no counterpart is present, no counter sites, means what? Eagerly it won't be able to go and make the bonds, and if the bonds are not being made, what will happen? It is of no use, because you can't stick the things up, you can't stick the other DNA part into it. Means a recombination won't be formed, and that creates what? Our issue. With this, the topic of this is over. Coming up to the next part, next enzymes. What are they? They are the synthesizing enzymes. Okay, the third one are the synthesizing enzymes. They will help in what? They will help in synthesizing the DNA fragment. Now you know that you will get what? You will get little bit amount of DNA, and from where you are getting the DNA of your interest, you either is making with the help of what? You are making with the DNA, so you will use DNA polymerase to make a copy of it, replication. What if you are not getting that? You are getting a RNA. From RNA also you can make DNA because of using what? The reverse transcriptase enzyme. So this is the case. Okay, so how we will synthesize? So these enzymes are used to synthesize new strand of DNA which is complementary to existing DNA or RNA, complementary to existing DNA or RNA template. So these could be of two types. One enzyme is what? Reverse transcriptase, and another one is what? Another one is the DNA polymerase. Reverse transcriptase, it makes RNA over DNA through this enzyme, and it makes DNA over DNA. That's it. Okay, so this is the synthesizing enzymes. After that, the last thing is what? Presence of ligase enzyme or the joining enzymes. So joining enzymes, this is called as molecular glues or what? Ligase enzyme. So they help in what? They help in joining the fragments of the DNA. You know that you will make a one fragment, means gene of interest and the vector. So these fragment has to be joined together. How it will be joined? With the help of these enzymes. So helps in joining the, you already know about DNA ligase from the molecular basis if you have following my channel. Okay, help in joining the DNA fragments together. Okay, with this your enzyme is over. The next comes the vehicle DNA. Okay, so next thing is second tool, that is vehicle DNA. It is also called as what? Vector DNA. Just remember the name. Now vector word we have studied before also. Similar kind of function will be here also. Okay, so it is what? DNA used as a carrier, like carrier of disease like mosquito, same over the carrier of the gene of interest. Okay, for transferring of foreign DNA. Means what? Your jellyfish DNA, okay, foreign DNA into a desirable host, Now normally plasmid and bacteriophage, they act naturally as the ability to replicate themselves and goes and act like what? As a transfer of the things. So we will be using them only. You know that they transfer the genes. Okay, so plasmids and bacteriophage have the ability to replicate, self-replicate within the bacterial cell. Now you know that plasmids are what? Plasmids are the one of the part of the bacteria. Okay, replicate within the bacterial cell independently of the control of chromosomal DNA. Now we're going to be understanding the examples of it. The first example is plasmid only. Examples. Number one is plasmid. Now when it comes to the plasmid, you have to remember that plasmid is what? It's a circular double-stranded DNA fragment, okay, present in the bacteria. So it is what? It's kind of an organelle part of the bacteria only, and it gives what? A special characteristics to the bacteria. Maybe it is could be fertility plasmid, it could be replicating plasmids, or so on, or it could be antibiotic plasmids. So this is what? This is a special characteristic these plasmids are providing, because they have what? Genes present on it, and you know that genes will give certain characteristics. They are just the genetic material only, but not the part of the major chromosomal material. Chromosomal material can replicate itself. So does they can also replicate themselves. Means that machinery is present on this plasmid also. Very important fact to remember and understand. Okay, so plasmids are there. For example, in our syllabus, PBR 322 plasmids from the E. coli bacteria, sorry, Salmonella typhi is there with us. Okay, so example. So plasmids are what? They are double-stranded, circular, chromosomal fragment, chromosomal DNA fragment found in bacteria which can self-replicate independently. Okay, so example PBR 322. Let's just try to make this one. Okay, So this is double strand, first strand, second strand. Of course, it won't look like this. It will look like what? It will look something like this. Okay, so means it has a outer ring and a inner ring. Okay, but since it is what, one over another, this area we can't see it at this level, right? We can't see it. We will see that it is some circle kind of a thing. So I have shown you that there is a certain things present on this. This part has certain thing. Okay, that's why I am making it like this. Now on this plasmid, so we are making what? PB322 plasmid. It has, first of all, we know that it can self-replicate. So there are certain self-replicating things would be present. So what it is, it has this gene present over here. This gene name is what? ORI. That is what? Origin of replication. Okay. Then there is what? There is replicating of protein, ROP present. Then there are what? There are many genes present over it, but two most important genes which is present on PBR322 is that antibody resistance genes. Means this plasmid, wherever bacteria it will be present, it will be what? It will give a feature of antibody resistance. Means that bacteria will not be die in when it is exposed to that medicine. Okay, so for example, here I am making that only. So this is one such gene and this is another such genes. So I made the two genes which are present on this. Clear? This is what? This is the tetracycline gene. Okay, so this is tetracycline resistant gene, whereas this yellow color which I have shown is the ampicillin resistant gene. Now these genes would be what? It will be very useful for us. And this is the plasmid. So plasmid also we have to cut, and we will cut it at this resistant site. Now think, if you have certain thing and I will cut it, will that thing will left with you? No sir, it is destroyed. You have cut it off, right? Same thing happens with it. The endonuclease, when it will cut at any part or anything or any gene, it will lose that feature. And certain endonucleases we know, whether it is endonuclease or exonuclease, but the cleaving enzymes we know. Okay, for now. Okay, so let's see that. First of all, I am writing the endonucleases, or sorry, first of all, I'm writing the nucleases enzyme which will be cutting the tetracycline. So two of them is there. This area can be cutted by BAM H1. Okay, simultaneously this area can be cutted by SAL1. Okay, in SAL1, S is for what? For the genus. AL is for what? The species. But there is a no strain written because strain is what? Optional fourth letter. And 1 is what? The first sequence obtained from this. Okay. Then over here, this ampicillin resistance, can you see? Over here, it can get a cut over here with the PSU1. Sorry, PVU. PVU1, and this part can be cutted with the PST1. Okay, then we have two more things that is also can cut. This is over here, this part, but here no antibodies are there, but it will come. What is what? EcoR1. So can we say that at this area, this GAATTC thing, palindromic sequence, will be present? Present. Okay, so this is how we're gonna be using the facts and understanding and knowledge what we have studied and try to put it over here. Over here we have what? HIND3 enzyme. This is a third enzyme that is turned out to be what? Endonuclease or exonuclease. What it is? from the hemophilia influenza. Understood this thing? We understood this point over here. Very important for you. Okay, now plasmid carries or carrying, not carries, carrying DNA. Which DNA? Alien DNA, means our gene of interest. Okay, integrated to it. Okay, so we added that thing. Now it is called as what? Recombinant DNA. Remember I have told you that a DNA with the foreign DNA means a new DNA is called recombinant DNA. It is also called as hybrid DNA. It is also called as chimeric DNA. Okay, or the plasmids. Okay, so integrated to it is known as recombinant plasmid, or what? Hybrid plasmid, or what? Chimeric plasmid. Chimeric plasmid. So these three names are being given. Except it, there is what? Ti plasmid is there, Ri plasmid is there, which is used in what? In the plant-based biotechnology. Okay, so it is used to transfer gene in plants. Which plants? Dicot plant mostly. This is what? This is the plasmid. Second example, who can act like a vector, is the viruses. So you know that bacteriophages are there. They can easily release the things. So the same thing is being talking. So we have talked already that plasmid and bacteriophage will be doing it. This we are doing it integrated form. So in this only example, you have to remember like lambda bacteriophage, which will give it what? To the bacteria, and retrovirus, which will give it to the eukaryotic cell. Okay, so this is to the prokaryotic cell, and this can be used for the eukaryotic cell. Now, this is the vectors we talked about. Now, the vectors must able to have certain features present to it. What features would be there? Okay, so three features we have to remember, and if we understand this, then we are able to understand the chapter's most important crux. What are they? So let's see the features that are required to facilitate cloning into a vector. Okay, so number one, A is ORI, that is origin of replication. Origin of replication will do what? It will initiate the self-replication of the plasmid. Means 1 to 2, 2 to 3, 2 to 4, 5, 6, something like that. It will keep on replicating. So these sequences are important because we want that it should self-replicate. So the one cell, when the cell in which it has present, it goes to the next, that feature is also there. Okay, so we understood this easiest point, that is sequence from where, what it is? Sequence from where replication starts. Then any piece of DNA linked to the vector having ORI can made to replicate within the host cell. Of course, because if this thing is present in the vector, it can replicate whether we are giving. So means if I put PBR322 into a bacteria cells or into my cell, let's take it an hypothetical example that it is possible that I put it in my cells, all my cell has that. Now what will happen if you will put me in the ampicillin or tetracycline resistant in the tetracycline or the ampicillin environment, I will resist to die. Imagine that I'm a bacteria only who can die in it normally. Okay, then comes what this is plus it is also responsible for controlling the number of copies it will make. Okay, controlling the copy number of the linked DNA. Whatever the DNA will put it, how many number of copies you want to make, it will do. Okay, second is very important, selectable markers, whose exact mechanism we will study later, but here we will understand certain basic of it. Okay, selectable markers. Now there are certain genes that should be there which you able to detect that whether it's present or not present can help them identified. So for example, if my body has taken PBR322, it means that my body should have a resistance towards the bacteria, that particular things which are present on that gene, right? So that thing able to find out is only possible if I put my whole body into the tetracycline or into the ampicillin environment, where ideally I should die. That never happens. It's a hypothetical situation. But since I have that plasmid present, I won't be dying. Okay. So this is what here we have to remember, that selectable marker helps in identifying and eliminating non-transformant from transformants. So means those who will be able to take this will be called as transformant. Those who are not able to take it called as non-transformant, and we can able to distinguish between them. It helps in that. And this will what? It will selectively permit and selectively permitting the growth of the transformant. Example, genes coding for resistance of antibiotic. How it works, we will do it in the next video. Okay, resistance to antibiotics. Okay, such as tetracycline, ampicillin. Okay, how does do it, we will do it later on. Here just understand certain point in your mind that it will come back again. Next is the restriction sites. Restriction sites means those sites which can be help in cutting it, because if we can't cut the vector DNA, how we will add the gene of interest. Okay, so restriction sites, or it is called as what? Cloning site. So this is what the vector needs recognition sites for commonly used restriction enzyme. Commonly used restriction enzymes. And at this site only we have to ligate, means we have to join the alien DNA, and it must to be carried out in any of the antibiotic site. For now we will remember that. Okay, so the ligation with respect to PBR322. Okay, the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. Okay, this is all about what your vector DNA. Next comes the third, that is the desirable DNA. Okay, so what you need? A desirable DNA. And how you can make it? You can directly make it over the DNA, or you can synthesize it over the DNA. Okay, so the third tool is what? Your desired DNA. This desired DNA is also called as what? It is called as passenger DNA, because it will be present on what? Over the vehicle. So this is what feels like passenger DNA. Alien DNA, because it is the outside DNA which we are putting it into another organism which is normally don't have that feature. Like the goldfish is never glow in dark, but it will glow in the dark. So we call it what? Alien DNA or the foreign DNA. Okay, this is all the other names which can be used as to refer it. Okay, so it is what? It is the DNA which is transferred from one organism into another by combining it with the vehicle DNA. And this complementary DNA or synthetic DNA can be made over the RNA or the DNA. So complementary DNA, which is called as cDNA, made over mRNA with the help of reverse transcriptase. Okay, with help of reverse transcriptase. Whereas a synthetic DNA called as sDNA is made over DNA through DNA polymerase enzyme. Understood? We understood. Then comes the last thing. Last tool is what? Host cell, in which we're gonna be doing what? We're gonna be putting up this thing. So last is the host cell. In host cell, it could be plant, bacteria, human, anything can be it. Okay, animal cells. So this is what? Cell used for DNA cloning or showing effect. Okay, which can host it inside its body. Okay, it can be plant cell, animal cell, bacterial cell, or fungal cell. Anyone which can be act like a host to us we will be using it up. Now that's all about the tools used in the biotechnology. Now, if you understand something about it, so material we has, the concept we little bit understood, we're gonna be elaborating it in the next video into simple 8 steps. And I assure you that way, if you will learn it, you will always remember it. My teacher taught me, now I'm gonna teach you. So let's just wait for it. If you like this video, give it a like, share it with your friends. Don't forget to subscribe, because you are going without even subscribing my channel. And you can even save this video for studying later on. I am coming up with a very good courses for all of you. So if you need one-on-one consultations, if you want one-on-one studies with me, your doubts to be solved, your centum is guaranteed, means 100 out of 100 marks is guaranteed, or you want to get 360 in NEET, you can definitely take up my course, which is going to be starting from the October itself. And all the details you can get on my Instagram in that chlorophyll underscore classes, which you can type it out on the Instagram and you can find it out very easily. And we even have certain cool stuffs going on Instagram. So why waste your time on stalking your ex when you can actually help, when you can actually study also? So that's all for this video. Thank you.